Hello everyone and welcome to Instant Biology by Dr. Neela. The topic that we would deal today is gram staining. Okay. So what is the use of gram staining? Why it is used? What is the method that is employed? What is the principle behind gram staining? We would discuss all of this, and apart from this we would also go on to discuss the differences between gram positive and gram negative bacteria, right? So let us first of all understand that what is gram staining. So gram staining is a method that is differential. What does differential mean? Differential means it can differentiate two things, right? So what can gram staining differentiate? Gram staining can differentiate the bacteria into two populations, that is gram positive and gram negative, right? So this gram positive and gram negative bacteria staining is due to a structural difference between both of these bacteria. So in which component this structural difference is present? It is present in cell wall. Okay. So cell wall is the entity due to which differential staining exists, or differential staining occurs in when we stain bacteria with gram staining. Why? Because the cell wall of the cell wall of gram positive bacteria behaves in a different way on staining with different dyes, and the cell wall of gram negative bacteria uh, behave in a different way or take a different dye color and on staining with gram staining, right? So this is the important difference, or this is the important basic on which gram staining is based upon, right? So let us now discuss the principle, or let us now discuss about the procedure of how gram staining is done, right? So first of all, the what do you have? You have a slide, right? What do you how do you prepare a slurry? You know that how a slurry or how a bacteria solution is prepared. You take a slide. Suppose this is your slide, and what do? You prepare a bacteria solution in which bacteria are present, and you drop a you pipette down a bacteria solution, a drop or so, and make it homogeneous around the slide. And then what do? You heat fix it, right? What is the meaning of heat fixation? With the help of heat, with the help of warmth, what happens? This bacterial solution is fixed upon onto the slide. Now no further movement or motility of this bacteria or motility of this solution from the slide won't be possible. So this is why this fixation needs to be done before performing the gram staining. Okay, so now we would move on to our first stain. Our first stain would be known as a primary stain, right? Because it is first of all used, then therefore it is called as a primary stain. Now what is our primary stain? Let us talk about that. Primary stain is crystal violet. So crystal violet is the primary stain. Its color is blue or violet in color. It is blue or violet in color. Now what do you do that after fixation of the bacteria onto the surface of the slide? You take the slide away from the heat, and then what do? You pour some drops of crystal violet, or you stain the slide with crystal violet. Now what happens actually the crystal violet, in case of gram negative, and gram positive, both of these bacteria, crystal violet would enter inside the cell, right? So let us understand this basic first of all, let us understand this basic cell wall structure of both gram positive and gram negative bacteria. In case of gram positive bacteria, this is the cell membrane, this portion is the cell membrane, and then we have lipoticoic acid, that has been shown over here in the form of blue lines. This lipoticoic acid is joined or is attached to the cell membrane, and this is followed by the peptidoglycan layer. This peptidoglycan layer is very thick in case of gram positive bacteria. How thick it is? It is around 60 layers. So 60 layers of peptidoglycan is present in case of gram positive bacteria, giving it a much compact structure, right? So this you can see that is a much compact structure, not much gaps are found in case of gram positive bacteria. Let us just go through this again. This is the cell membrane. You would be knowing that this is the phosphate end and this is the lipid end, and that is present over here. Cell membrane, this portion is a cell membrane. It is followed by lipoticoic acid, and then we move on to peptidoglycan. So phospholipid bilayer, you might be knowing this is the cell membrane that is common in both the gram positive and gram negative bacteria. Now coming to the gram negative bacteria, having discussed gram positive bacteria, let us discuss gram negative bacteria. The cell membrane is the same. This is the phospholipid bilayer. You can see over here we have mentioned it as inner membrane. This is followed by periplasmic space, right? This is the periplasmic space. Nothing is present over here. Actually, beta lactamase enzymes are present over here, but that is not a part of the lecture of our current lecture. We would discuss it in some other lecture when we would deal with the effect of penicillin on gram negative bacteria. So this is the periplasmic space. This again is followed by peptidoglycan. This is peptidoglycan layer. Let me write it over here. So this is the peptidoglycan layer. You can see a difference between the peptidoglycan layer present over here and which is present over here. What is the difference? The difference is very evident, and it is that here the peptidoglycan thickness is very less as compared to the thickness that is present over here. Here 60 layers are present, and here only around 3 layers are found, right? So you can see the difference between the peptidoglycan thickness that is present in case of gram positive and gram negative bacteria. Okay, now let us move ahead and discuss that what is above the peptidoglycan layer. Above the peptidoglycan layer is a outer membrane, right? It again is of the same structure as that of the inner membrane. It has a phospholipid bilayer, but it also has lipopolysaccharide. Okay, you might be knowing about lipopolysaccharides, that those are also sometimes endotoxins, and they are complexes of lipids and polysaccharides. Polysaccharides contain the O antigen. You would be knowing that. Let's not discuss about this. Let's just take an overview about gram negative cell wall. So it has inner membrane and outer membrane, which are separated by peptidoglycan layer and periplasmic space. So this is the entire structural overview about gram positive and gram negative bacteria. I hope this would be clear to all of you. Now let us come back to what we were discussing a little while ago. We were discussing that the first or the primary stain. Okay, let me write over here. So this is our primary stain. So crystal violet turns the cell into a blue color. Okay, when you use crystal violet, the crystal violet would pass right through the peptidoglycan layer, cell membrane, and go inside the cell and stain it in a blue color or violet color in case of gram positive bacteria. And similarly, in case of gram negative bacteria also, what will happen? The crystal violet will go through the outer membrane, through the peptidoglycan, through the periplasmic space and inner membrane into the cell and stain it into a crystal violet, or stain it into the violet or blue color, right? So this would be the effect of crystal violet, right? This is the primary stain. Remember, the primary stain in gram staining is crystal violet. This is often asked in examination as multiple choice question. Now coming to the second enzyme, the second chemical that we use. The second chemical is also called as mordant, and it is iodine. Okay, right? So iodine is used as a mordant. Now let us understand what is mordant. Mordant, the function of mordant is to fix the primary stain inside the cell itself. Okay, let us understand this once again. The function of mordant is to fix the primary stain inside the cell. Okay, so what happens is basically the cell, sorry, the primary stain, suppose this is the primary stain and this is the iodine, right? So this primary stain and this iodine interact with one another and form a complex, this which is called as a CVI complex. CVI complex. What does CVI stand for? Crystal violet iodine complex. Okay, so this is formed after the iodine is used as a mordant, and iodine also goes inside the cell and forms a complex with crystal violet. Okay, so up till now what has happened? What has happened is we have stained our cell with crystal violet. Let us assume that just after staining with crystal violet, you see both gram positive and gram negative bacteria. What will happen? Both of them would look violet in color or blue in color, right? Just after staining with iodine, what will happen? Crystal violet would form a complex with iodine, CVI complex that is called, and the color would be same as violet. Now one thing important, or one important occurrence would take place, that would be after the formation of CVI complex, the structure of this complex would increase in size to such an extent that it would be difficult for this CVI complex to pass away from, or to pass out from the gram positive and gram negative bacteria just now, just for this purpose, just for instantaneously. This CVI complex would not be able to come out of the cell wall of both these gram positive and gram negative bacteria. If the function of CVI complex is to just fix the primary stain inside the cell. Okay, so it would increase in size and the stain would stay inside the cell in both gram positive and gram negative bacteria. So up till now, I guess this would have been very clear to you. And how does crystal violet pass inside the cell? Because crystal violet is very small molecule that can easily pass through the cell, but after joining with crystal violet, it forms a bigger complex that would not be able to pass through the cell, outside the cell, very easily. Okay, so this was all about mordant. Now the third chemical that we would be using is alcohol. So what is the function of alcohol? Now this is very important. We should understand this with much attention. Alcohol. So you might be knowing that the function of alcohol is it acts as a dehydrating agent, which means that it would pull off all the water molecules that are present inside a tissue, inside a cell. And one another thing that it performs is it dissolves all the lipid components that are present inside a cell or present in the membrane. Okay, so let us understand this alcohol. What does it do? It acts as a dehydrating agent, and also it acts as a it dissolves the lipids. It dissolves the lipid moiety, right? So once you put alcohol on these cells, that is gram positive and gram negative cells, what will happen is alcohol here would be in contact with only peptidoglycan, right? Therefore, not much effect would be seen on the gram positive bacteria. The only effect that would be seen on gram positive bacteria is that the peptidoglycan layer would lose water suppose this is the peptidoglycan layer. This would lose water and it would become much more compact, right? It would become much more compact after treating with alcohol. Okay, this would be in case of gram positive bacteria. Okay, so now no effect of dissolving of lipid would take place because we cannot see any lipid in this portion or the outermost area, outermost surface of gram positive bacteria. But here, in case of gram negative bacteria, we can see this lipopolysaccharide. This has a lipid component. The outer membrane also has this phospholipid bilayer. This entire thing is comprises of lipids, right? So this entire thing would be wiped off. Okay, assume now that this portion is not here and only peptidoglycan layer is there, periplasmic space is there, and inner membrane is there after treatment with the alcohol. Okay, so this would be the case in case of gram negative bacteria. Only a thin peptidoglycan layer would be present outside and all this portion would be wiped off due to the treatment with alcohol, right? So now just assume or just understand this, that after treatment of alcohol, when this peptidoglycan layer would become more compact, now the crystal violet would not be able to pass out, that the crystal violet that we had used earlier would not be able to pass out from the cell, and now if we try and visualize this gram positive cell, it would be seen as violet or blue in color. But if you try and visualize gram negative bacteria, then what will happen is, because this complete portion would be wiped out, and after the treatment with alcohol, peptidoglycan layer is not very thick, which can withstand the action of alcohol. This would create some pores, or it would not be able to handle, or it would not be able to fix, or it would not be able to resist the passing out of the crystal violet. Okay, so what will happen in this case, gram negative bacteria case, due to the thin peptidoglycan layer, what will happen, the crystal violet would not be able to be present inside the cell, it would go out of the cell. Okay, so after this step, if you try and visualize gram negative cell, how would it look like? It would look like unstained cell, it would have no color, right? So why will this happen? This will happen because this portion would be completely wiped out, and peptidoglycan has not that much of strength to withstand the action of alcohol, and the crystal violet will therefore, as consequently, would come out from this layer and move out of the cell, and resultant of this procedure would be colorless gram negative cells. Okay, and what would be the color of gram positive? It would continue to be as violet or blue in color, right? So this was the effect of alcohol. Alcohol acts as a dehydrating agent, as well as it acts in the dissolution of lipids. So after the action of alcohol, the next action would be of the next chemical that we would use would be secondary string, and that would be safranin. So how does safranin look like? It look like it stains the cell in a red or pinkish color. So when you stain with safranin, in case of gram positive bacteria, the peptidoglycan is tight enough. Okay, so it won't generally allow the safranin to go inside, and if in case the safranin goes inside also, what will happen? The effect or the color of safranin would be masked due to the presence of the violet color or the dark bluish color of crystal violet which was present beforehand only. So firstly, it won't pass only, and secondly, if some amount of stain passes inside, then also the color of red or pink would be masked due to the presence of violet color of gram positive cells. So gram positive cells would look like violet only just after seeing after the safranin staining. If you see the gram positive cells inside the microscope, you would see violet color only. Okay, but once you stain the gram negative cells with safranin, what will happen is, because this peptidoglycan was already weak, these safranin molecules would go inside and stain the cell pinkish or reddish in color. Therefore, after the after the staining with safranin, if you try and visualize the gram negative cells, they would look pink in color or red in color. So that is why we call it differential staining, because it can differentiate the gram positive bacteria and gram negative bacteria on the basis of the stain which they take. Gram positive will take in crystal violet and then therefore would look violet or blue in color, and the gram negative stain would take in pink or reddish color of safranin due to the change in the structure of cell wall in both of these cases. Peptidoglycan is thick in case of gram positive bacteria and thin in case of gram negative bacteria. That is one of the major reasons why this kind of differentiation takes place, right? So this was about the process, how this staining is performed. Now let us come to the differences between the gram positive and gram negative bacteria. I have employed or I have used some points on which we would differentiate gram positive and gram negative bacteria. So first of all, the first thing that we can take in here is the gram reaction. Okay, so what is the result of gram reaction in case of gram positive bacteria and gram negative bacteria? If we talk about the gram reaction, in case of gram reaction, gram positive bacteria would stain, would take up the primary stain. That means they would look violet or blue, whereas here they would look gram negative, they would look red or pink. Right, now let us come to peptidoglycan layer. The peptidoglycan layer is very thick in case of gram positive bacteria and thin in case of gram negative bacteria. Now teichoic acid, you can see that lipoteichoic acid is present in gram positive bacteria, but this is absent in gram negative bacteria. Now periplasmic space, again periplasmic space is absent in gram positive bacteria and present in gram negative bacteria, right? Outer membrane, now you cannot see outer membrane over here, which was being washed away by alcohol. So outer membrane is only present in gram positive bacteria and is not present in is only present in gram negative bacteria and is not present in gram positive bacteria, right? Now coming to lipid and lipoprotein content. Lipid and lipoprotein content, lipid content is more here. Lipid content is high over here, that is gram negative bacteria. Lipid content is high and it is low in case of gram positive bacteria. So let us also take examples of gram positive and gram negative bacteria. In case of gram positive bacteria, examples can be Bacillus, and in case of gram negative bacteria, Enterobacter can be suitable examples. So this was all about gram staining. If you liked my lecture, please click the like button, and if you please subscribe to my channel, and if you have any comments, please write it on the comment section. I would try and resolve them in the upcoming videos. Thank you so much.